All right, this is the syllabus for this class. And we are meeting on Tuesdays and Thursdays at 3:30. And there's a Zoom link in the syllabus. And it will also be available on CCLE once we have the websites merged. And so currently I spend my office hour on Tuesdays at 2:15 to 3:15 right before this class. But if this time won't work for you, you can schedule an appointment with me. So I'm going to send a link for this office hour later, and that will also be posted on CCLE. So an introduction to this course, I'm going to introduce statistical and computational methods that are applicable to computational biology and bioinformatics. And I will emphasize on the understanding of basic statistical concepts and the ability to use statistical inference to solve biological problems. So we will see the definition of statistical inference soon. And this course will cover a wide range of statistical methods, including statistical inference, which covers hypothesis testing, confidence intervals, likelihood-based inference, and multiple testing correction. Unsupervised learning that includes clustering, dimension reduction, and measures of association. And supervised learning, including regression and classification. And sampling methods will focus on the Markov chain Monte Carlo for sampling methods. So the prerequisites I listed here are our undergraduate courses offered in my department at UCLA, including Stats 100B, that's about probability, and Stats 100A about probability, B about mathematical statistics. If you didn't do your undergrad here, you will find equivalent classes in your home university. If you don't have the prerequisite but still enroll in this class, I will strongly suggest that you make up those background materials during your leisure time so that you will not feel this class is too dense for you or too advanced for you. And if you want to have access to the recordings of videos available and made by my colleagues for those two courses, you can send me an email and I can refer you to my colleague and he can send you links or CCLE links so you can watch his video recordings. I think that would be a good way to make up the background. And I know some of our bioinformatics students have enrolled in Stats 100B based on my suggestion. So you will see that the materials are really useful. For example, with that class, you will understand what is a T distribution, what is a chi-square distribution, what is likelihood, right? What is the maximum likelihood estimates. So those things will be used in this class. And we don't have a textbook. So I'm currently writing the textbook based on my teaching experience for this course, but it's not ready yet. So I will post my lecture notes on CCLE, and I will also post reference papers to CCLE for your further reading. So those are the optional materials for those of you who are interested in knowing more. And our course grades are based on homework assignments. They will be bi-weekly. I anticipate that there will be 3 bi-weekly homework assignments and will be due in 2 weeks. So because we are doing this remote teaching, every homework will be submitted electronically. So please try to compile your homework files into one file, including your writing, your answers, and your programming code, so that our reader, special reader, will grade your homework by referring to your code if they need. And for the project, that's the second part for this course. So I will encourage you to form groups with no more than 2 students per group. So in other words, if you prefer to work by yourself, that's fine. You can just have your own group. But given that our class is relatively not so small, so if we have fewer groups, then we can probably do a longer presentation in the last week, right? So currently I set it to be 10 minutes, but if it turns out that we have more groups, we may shorten the presentation time. So we will ask for your group formulation, or we'll ask for your groups, and maybe in week 7, I would say, so that we can know how many groups are there. And for this project, I will assign a data set to everyone. So that will be given to you in the week 4, so that everyone will have the same data set to work on. But you can formulate the project idea by yourself. It could either be a biological question, like what answers do you want to get out of the data set, or a statistical question. If you want to try some methods on this data set and see their performance, that's also fine, or both. And I would encourage that for the groups, it would be better if we have one student from more of a statistical background, another student from a biological background. Then you can combine your strength into this project. And in the past years, we have seen a lot of interesting projects, and some groups even had their projects published in the end. So that's quite encouraging. So this can make sure that in the final presentation, everyone can understand each other's projects. And that also you can learn interesting ideas from your peers. Okay, so then you will answer the questions by using but not limited to the statistical methods we cover in this course. You may come to my office hours to discuss about your project. And my suggestion is to start early. Don't wait until the last minute to do the project. You will have the data set since week 4. So you have about 5 weeks to work on your project before your oral presentation in week 10. And then you have 1 more week to write down your paper, the project report. That will be due by the Sunday of the final week. So for your information, this paper will have a format requirement. So we require the total length to be no longer than 6 pages, but this excludes figures, tables, or references. And the font size 12 point, single state. So we can make sure that every group has approximately the same amount of materials in the report. So the grades will be based on 40% homework, 20% the final week presentation, and 40% for the paper. So I actually set the oral presentation to be quite some percentage, because I think for bioinformatics students or students in biosciences in general, the speaking or giving presentation is a very important skill. So you will need to practice this a lot. And for the group, if you have 2 people in your group, so usually people do the oral presentation together. And also in the final paper, you need to specify the contribution by each group member. Is it 50-50, 40-60? So we can know each one's contribution. And so you don't need to worry that I did most of the work, but my peer didn't contribute much. That's not gonna happen. Okay, so this is the course schedule I listed here. So we'll see how it goes. We will roughly follow this schedule, but if we are slower or faster, we will adjust the schedule accordingly. Okay, and as I said, lecture notes will be posted. So the course policy, the first one, the only one I would say is to encourage everyone, if you're attending this class live, try to turn on your video. I think the benefit will be twofold. For me, I will see you guys and I can feel, okay, I'm talking to real people, not just my computer screen. And for you guys, I feel like having your video on is probably a good way to make you concentrate better, but we'll see. But if you do have concerns about like your environment, etc., you can let me know and you can turn your video. If you cannot make the live sessions, you can watch the video recording. I'll post them to YouTube, so that will not be a concern. Okay, so I think that's it. The last thing, please keep your email address current at myucla.edu so you can receive my class announcement. Otherwise, you will miss the announcement. So if you didn't receive announcements yet, like the one I just sent a few minutes ago about the Zoom link, you should check your email address to make sure that you have the correct email on file. So that's what I want to say about the syllabus. Do we have questions about the syllabus before we move on to having you guys introduce yourselves? Okay, seems very good. So for the introduction, my suggestion is that since we cannot see each other in person, and this will be like our first time seeing each other for most of you, maybe I think the introduction, I would like to include have everyone say what's your current major, what year are you in? Like you can say I'm a 4th year PhD student in bioinformatics, for example, and then what's the reason that what's your statistical background? So how many courses have you taken before, and what's your motivation for taking this class? What do you want to get out of this class? So that can help me understand your expectations. Probably we can start from the name list I see on my screen. So the first one, Albert Xue.
Hello.
Hello. I think you're muted. We cannot hear you.
Hello? Hello?
Hello? Hello? Oh, I think, okay, that's fine. So Albert, maybe you can just type your introduction in the chat box. Okay, no problem. We'll just continue. Angela?
Hi, so I'm a first year graduate student in bioinformatics. I was a math major and I had a stats minor in undergrad. I'm originally from Kentucky. I went to the University of Kentucky, so I've taken some of the more applied stats courses and some basic inference and probability. On why I'm taking this course, I'd be interested to get more familiar with machine learning and the technical and the theoretical aspects of it.
Okay, thank you. Welcome, April.
Hello.
Hello.
Oh, okay, it's working. Hi. So I turned on my webcam, but it's on my phone and I've been having trouble with that. So I'll just go ahead and introduce myself anyways. I'm April. I'm a 5th year in the Computational Systems Bio BSMS program.
Okay.
So taking this class is going to be a little bit challenging for myself, but I'm interested in learning more statistical methods.
Okay. Okay, welcome. And Albert, can you speak now?
Hi, can you hear me?
Yes, I can hear you now.
Okay, good. I'm Albert. I'm a first year bioinformatics student. In undergrad, I did math CS, but I sort of avoided stats classes because they were all at 8:30 and I didn't really like stats. So I guess my motivation for taking this course is to sort of catch up on that sort of stuff and apply that to my research.
Okay, thank you. Hopefully you will find this class more interesting. But I would say that for bioinformatics students, many of you take this class probably just because it's in your core curriculum. So you have to take it, right? Or otherwise you have to find a reason to not take it. But I think for other major students from other major, probably that question is more reasonable. What motivated you to take this class? Yeah, thank you. And Carolyn.
Hey everyone, I'm Caroline. I'm a first year bioinformatics PhD student. My bioinformatics background has pretty much been nothing until last quarter, so this has been really fun. So I'm hoping this is, to be honest, one of my required courses, but I definitely will learn a lot because I have not a lot of foundation, so it'll be good.
Okay, thank you, Carolyn, and welcome to UCLA. And our next student is Daniel Vo.
Hi, I'm Daniel. I'm a 4th year doing computational bio and the BSMS program with bioinformatics. And this is also part of my core courses for that. This isn't the first bioinformatics course that I've taken. And so I'm looking forward to this.
Okay, thank you. Yeah, it seems that most of our students, based on the ones who spoke just now, the majority of them are either first year bioinformatics PhD students or that BSMS computational and systems biology major, right? So, okay. So next one, Diana Zhang.
I'm Diana. I'm a 2nd year in the Stats MS program, and I have a background in Stats and CS from UC Berkeley before this. I haven't done anything with computational biology, and it just sounds like something cool to learn.
Okay, great. Yeah, so in this case, I think it would be good if you can find a partner who have a biological background for that final project. So next one, Eileen Huang.
Hi, I'm Elaine. I'm a first year bioinformatics student. My stats background is from taking Stats 100B last quarter. And other than my motivation in this class is I think it would be great if I could better understand the statistical methods and papers when I'm reading them for my research.
Okay, great. welcome. And so, yeah, maybe this is already a good opportunity for you to think about your possible partners, since we have everyone here. And next one is Gabe Hassler.
Hi, I'm Gabe. I'm a fourth year biomathematics PhD student. And my research focuses on developing statistical methods for phylogenetics. I'm taking this class because I know a lot about kind of my very narrow area of statistics, but I'd like to get a kind of broader sense of what the current state of things is.
Okay, welcome. And Harold.
Hey everyone, I'm Harold. I'm a first year PhD in the bioinformatics program. In terms of my statistical background, I've taken like 45 stats courses back when I was when I had my master's degree. I'm taking these courses because it's, first of all, it's required, and second of all, I just kind of want to see how more examples of statistical concepts being applied in bioinformatics research.
Okay, welcome. Next student is Hui Ling.
Hello, my name is Hui Ling. I'm also a first year bioinformatics PhD. I did the BS/MS, like Computational System Biology program here at UCLA. So I take most of the undergrad stats courses, and I've also taken one grad courses in biostatistics, which is about simulations. But I feel like most of the stats courses that I've taken are not like that bioinformatics related. So I feel like I've learned like a lot of different like topics, but like when I've used like DSeq2, but like I'm not that familiar with like how DSeq2 works. So I feel this course is really excited to like let me like feel the bridge between those like stats topics and like the real like bioinformatics applications.
That's something we aim to achieve. So hopefully we can achieve this together. So actually one thing, just as an introduction, one thing I want to focus on is we will talk about DSeq here actually. And one thing that's interesting to know is that DSeq2 differs quite a lot from DSeq. Actually, the statistical approach is almost new. And so then we will actually do a dissection of DSeq. And we had a lot of fun in the past years. I will ask the student to see what is not reasonable, what is reasonable in the DSeq method. And then we will actually be detectives together, try to see why those statistical methods make sense or do not make sense for the biological application. So that's something I want to try to do this together with you guys, so that then in the future, even though you're not a developer, method developer, when you use other people's method, at least you have more ways to see whether the method makes sense to you or not. Okay, so next student, Jamie.
Hi, I'm Jamie. I'm also a first year bioinformatics student. My background in statistics, like Elaine, is pretty much just stats 100B, or I learned that I didn't know as much stats as I thought I knew.
So actually, so may I ask, how do you feel about stats 100B in the last quarter?
It was, I mean, I think like one of the hardest parts was like having to remember math that you learned a while ago. Like I hadn't taken LinAlg in a while, and it showed.
But at least that's kind of a warm up. So then you will not complain too much about my course.
It was definitely a warm up. Yeah, so I'm a bioinformatics student, so we have to take this course. But I mean, I'm excited to kind of fill in gaps. I feel like I use a lot of statistical methods and applications, but then I feel like I'm missing on the theory. So I'm excited to fill in there.
Okay, welcome. And our next student is Jennifer.
Hi, I'm a first year human genetics student. I did a BS and MS at UCSD, and I've taken a fair number of bioinformatics and statistics courses, but I'm interested in this course because I just want to make sure I'm not missing anything and I'm filling in any gaps I have.
Okay, welcome. And next is Jingyuan.
Hi, everyone. My name is Jingyuan, and I'm currently a first year CS PhD student. And I was also a BS/MS in computational systems biology at UCLA. My statistical background is that like I also took like many stats courses that are like undergraduate stats courses when I was an undergrad. And last quarter I took stats 200A, and this quarter I'm also taking stats 200B. So like hopefully like I'm learning enough stats background already to like take this course, but I would also want to know like whether this course could help me better connect those stats concepts and methods into like research in bioinformatics.
Okay, yeah, thank you. And also, that's something I want to achieve. I understand that this class has a very diverse background. The students have different levels of statistical preparation. So my goal is to give some high level insights that everyone can hopefully understand. And for those of you who want to go deeper, I will provide materials for you to look into the details. But I'll try to explain concepts not using too much math. That's just my goal. But I don't know how well I can achieve. I'll just try to do that. So hopefully that it will not be so boring to you, and at least you can appreciate the importance of math. It's not just some boring stuff. It can really help you solve the problem. And next, John Ma. John.
Hi, how's it going? I'm John.
Hi. Hi.
I'm a first year bioinformatics PhD student. I don't really got like a formal background in stats. I only took one class, but my undergrad research was on statistical phylogenetics.
I see. That's quite statistical, actually, if you're working on statistical phylogenetics.
So I really like stats, but I'm looking forward to actually learning it because I haven't.
Okay. Okay, welcome. And Jonathan?
Hi, I'm Jonathan. I'm also a first year bioinformatics student and a survivor of Stat 100B. Previously, I double majored in computer science and statistics, but statistics was the lesser double major, and that was more than 4 years ago. So yeah, I'm looking forward to taking more stats courses at UCLA.
Okay, welcome. Oh, did you do your undergrad at UCLA as well?
Oh no, I'm from Canada and I did it in Canada.
I see. Yeah, based on the previous students' introduction, seems that UCLA has done a great job in retaining our own undergraduate students, right? We have several of you who did undergrad or master at UCLA and then continue as PhD students. Okay, Kimberly.
Hi guys, my name is Kim. So I'm actually a second year PhD student in the anthropology department, but I'll explain in a little bit why I'm here. Previously in undergrad, I took like applied stats and stuff like that, but in grad school, I've basically only taken Bayesian stats, and that was cool. It was super fun. But yeah, I'm here because I do computational genetics. I do epigenetics and high altitude adaptations for my PhD stuff. So I don't know. I'm here because I feel like I use a lot of programs that have like these beautiful statistical like models behind them, but I don't know how they work. I just kind of like take the developer's word for it that they do work, and that's not great. So I want to fix that.
Yeah, I totally understand. Yeah, that's the typical thing. Still, my students who are already like senior PhD students in statistics, sometimes they still feel like a lot of the bioinformatics methods, their statistical part are so messy or mysterious to understand. that's a common thing. Yeah, so we'll try to see. Yeah, we'll try to make things simpler. Next, Cody.
I'm a fourth year PhD student in computer science. I did statistics as an undergrad, and I feel like I like the theory and I do like research, but they don't really like touch each other. It's kind of the same thing as like when reading papers and stuff, like really understanding the theory. So I would like to mesh that more into each other.
Okay, yeah. Okay, welcome. And did we miss Gary when we did the introduction? Gary Z?
Hi, I'm Gary Zhou. I'm a second year biomath PhD student. I've only taken one stats course before, which is the mandatory core class for biomathematics.
What is that?
That's Biomath 205. I'm not sure. I don't remember the exact.
204.
204, yes, 204. Thank you.
So what did that cover in the core course?
Linear regression.
Yeah. Okay, I see. Okay, yeah, so that will still be quite useful because I know that in linear regression, people will talk about statistical inference. So that shouldn't be a new topic to you. But to be honest, I think inference is the most difficult part to understand for people who didn't take statistics classes. Okay, welcome, Gary. Next one is Mariana.
Hi, I'm Mariana. I'm a second year biomath PhD student. I was a applied math major in undergrad, and I took a few stats and probability courses, and I also took 204 from like the core biomath requirements. And I'm interested in getting more familiar with like machine learning methods in computational biology.
Yeah, I actually know that in the bioinformatics program, there's another class taught by Shriram. It's called the Machine Learning in Computational Biology. Yeah, so if you're interested, you may also check out that class. Okay, next one, let me see, Mark Xiang.
Hi guys, my name is Mark Xiang. I'm a first year bioinformatics student. As for the statistical background, I only have intro level statistic class for undergrad. Taking this course is one of my requirements. I wanted to catch up with statistics and also see some application to use it in the bioinformatics research study. Thank you.
Okay, welcome. Next one, Matthew.
Hi, everyone. My name is Matt. I'm actually a second year postdoc here at UCLA, and my background is in physiology, and the stats that we did there was very superficial, just t-tests and some ANOVAs, a little bit of linear regression. And for my postdoc, I'm doing some genomic analysis. And after reading for the last year, I really wanted to make sure that I had a handle on the stats that I was going to be using. Some of the analyses that we're doing, it sometimes seems like it's the wild, wild west, the way they're using some of these methods. And I just want to make sure that I understand these things properly when I'm applying them and get as much information as I can.
Okay, yeah, welcome. And I know that you are here to audit this class, right, as a postdoc. So your research experience will be very helpful. If you have any comments or questions during the class, please feel free to speak up. We have another Matt here in the class, Matthew Hatfield.
Yeah, hi, I'm Matthew. I'm another first year PhD student in bioinformatics. I really don't like stats, but to be fair, I haven't enjoyed any other subject until it gets mixed with biology. So this is the first stats class with biological applications I've taken. So I'm excited to see how that can get me driven to learn stats better.
Okay, well, welcome. So but do you like computation?
When it's related to biology, yes, I do.
Okay, yeah, otherwise you would not choose bioinformatics, right? You could choose a web lab biology.
Oh yeah, I do enjoy computational applications in biology and sometimes with other things.
Okay, welcome. Next one, Ran.
Hello, everyone. I'm a first year bioinformatics PhD student, and I had a few fundamental statistical course in my undergrad and also statistical method in machine learning, but I'm looking forward to learn more statistical applications, methods in bioinformatics, and hopefully it will help my own research. Thanks.
Hopefully. Okay, welcome. Next one, Samantha.
Hi, my name is Samantha, and I'm an environmental engineering PhD student. I also did my bachelor's in environmental engineering, so I had environmental statistics, and I've taken like few courses online and I really like statistics, which is why I'm pursuing my super minor in it as part of my PhD. And I took grad courses last quarter, one with you, Professor Lee, and with A as well. And they were a little dense.
Yeah, that one is much denser. I would say that one will be much denser than this one.
But like when we were doing the project and stuff, and I wanted to like kind of pursue what I was doing in environmental engineering and like kind of mash it up, I thought that I didn't get enough out of the paper, like all the reading that I was doing, and maybe that this course could actually help me in my own research better.
Okay, welcome. Nice to see you again, Samantha. And next one, Trevor. Yeah, he's also in my other class this quarter.
Hi, Dr. Lee, and
Hi.
hello, everyone. I'm Trevor. I'm yet another first-year bioinformatics PhD student. I got a bachelor's in bioinformatics at UC Santa Cruz, where I also minored in statistics. So I learned some machine learning theory and took some other classes as well. But I would say that Santa Cruz really emphasizes HMMs. I know they did like some of the Human Genome Project there. So they're just like over the top with HMMs and Bayesian statistics. So I feel like I have gaps everywhere else. So it was good to take the 100B class. I noticed like, you know, all my other members of my cohort here who survived. That class was tough. It was just like a consistently high workload. But I think it was a great review. And I would echo what Elaine said. You know, you read through these papers and you see these methods used, and like I'm often not sure why they use that method. So it'd be nice to learn that in this class.
Yeah, welcome. And yeah, I think here in bioinformatics, probably you will also learn HMM in some other bioinformatics courses. So I'm going to skip that. I'm not going to talk about HMM in this class, but we're going to talk about EM algorithm, which is the foundation of the parameter estimation in HMM. Okay, so maybe we can do the students first, and then we'll let our special reader to introduce himself. So yeah, you're next.
Hello, everyone. I'm a first-year bioinformatics PhD student, and actually, I don't have much background about mathematical or statistics because I'm from a biochemistry background, and this is also my motivation to enrolling these related courses. Actually, I want to enroll in a statistic 100B or 100A in this year, but I'm sorry they don't open to the bioinformatics students in this year. Yeah, so maybe
Yeah,
I want.
So in this case, you can ask for the video links to watch yourself if you want to make any background.
Okay, I think that would be much better.
Yeah. Yeah, I
Okay,
remember
thank
you gave the presentation in the 201 bioinformatics 201 seminar course about the nanopore sequencing, right? Yeah, so that's quite related to your biochemistry background, I would say.
Great. Okay,
Yeah.
thank you.
Yeah. Okay, welcome. Next one is Yu.
Hi everyone, my name is Yu and I'm a second year medical informatics PhD student. So I did my undergrad in biology, so I actually don't have that much stat background. But I did take two stat grad class at UCLA. But then I realized they are not that related to what my research. So that's the reason I want to enroll in this course because I think it's more directly related to what I will be studying on.
Okay. Okay, welcome. So we also have a visitor, Ze. Do you want to introduce yourself? He's from UC Irvine, and so because he's in my collaborators lab and he's interested in statistics, so he's auditing this class.
Hi, my name is Ze. I am a first year cellular molecular biology students, PhD students at UC Irvine. Before I joined the PhD program, during my undergrad, I was deliberate trying to avoid statistics class as much as possible because I had a very bad experience first year, freshman year for statistics class. I mean, I did well, but just the class is just way too complex in a way. But what funny is through the research experience that I had, I always get dragged back to statistics. So it's kind of like a fate. I can't really avoid it at all. I was cursed. So I actually did a ANOVA about statistics and also introduction into the linear regression, and yeah, and now I'm doing pretty much bioinformatic rotations, and happy to be here. Thanks.
Welcome. So this is the benefit of remote teaching. So if you're interested, you may find some resources outside your own school. And finally, let's have Wenbing introduce himself. He's the special reader for this class. He's a third-year bioinformatics student. So basically, he's 2 years above many of you guys in the bioinformatics program. So Wenbing, would you like to introduce yourself?
Oh, sure. Yeah, hi everyone. I'm Wenbing. And yeah, just as Jessica said, I'm a third-year student in bioinformatics. And I took this course perhaps 2 years ago. And I think it's a really useful class for many of you who want to learn the basic techniques in bioinformatics. For example, we will learn about the differential gene expression, and we will learn about cutting edge techniques such as clustering, and also many other fancy topics like the MCMC yeah, in statistics. So it's quite useful, and yeah, that's pretty much what I want to say. Good luck and have fun.
Thank you, Wenbing. Thank you for being here. Actually, it's not required that you attend the classes, but it will be nice to see you here. I think for all of us. Yeah. So thank you for the introduction. That's a nice way to see each other remotely. Hopefully, I can see you in person after we reopen the campus. Hopefully in the fall. So for today is going to be quite light, the lecture. And after that, today we're going to go into the technical materials. But for today, I want to give all of us some high-level introduction to what roles does statistics play in bioinformatics or biological research. So I'm going to show you some highlights from the papers I sent. I sent a Dropbox link via my UCLA. So you should be able to see the PDF because the CCLA is still not working, unfortunately. So this is a paper from Science written by 2 statisticians I know in Johns Hopkins, biostatisticians. So they wrote a lot of those, I would say, articles for the general biological audience, which I think is a very great contribution to the field. So they try to use the languages that are not so mathematical to introduce the key insights in statistical application. So the first thing I want everybody to think of before you do any statistical analysis is, what is the question? Okay, mistaken the type of question will actually lead to some analysis results that cannot answer your question. And this is a common mistake in many biological papers. They seemingly do some statistical analysis, but the analysis result do not reflect the answer to their scientific question. Such a misalignment can lead to wrong scientific conclusions. So I'm going to skip all the text. You can read this yourself after the class. But this is a graph I think is a key point of this paper. So this is called a data analysis flow chart. So I think it's a good way to walk through those steps when you design your statistical analysis. And I think for now, probably a lot of answers are not so clear to you, but you can revisit this chart after taking this class and see if you understand better. Okay, so the first thing is, did you summarize the data? If not, you're not doing a data analysis. I think this is trivial, right? If you're going to extract information from your data, then yes, you're doing some kind of summary, so you go yes. Okay, did you report the summaries without interpretation? That's called descriptive. And that's very common in a lot of seminar talks we hear. So for example, people may present the data in a chart, right? They say, okay, let me just make up one example. Like in the first seminar talk we heard this week. So the speaker talked about the different numbers of alleles, right? For each type of genomic region. You see that bar plot if you were there. But if you're not, you can still imagine what it's like. So say for exons, for some intergenic regions. So for each type of genomic regions, we have a bar to summarize and count. If we just summarize those counts without making interpretation, then it's called descriptive. And descriptive is not usually a good word in scientific research. If somebody says, okay, the paper is too descriptive, it means that the paper just presents some numbers without making interpretation. So that's not so good in scientific research. Okay, so we want to go down. Did you quantify whether your discoveries are likely to hold in a new sample? If no, you are just doing exploratory data analysis. It means that you're just exploring your current data, but you're not trying to look beyond your current data. And in our scientific research, especially in biology, we often want to make some conclusions that hold in general, not just for this particular data set. So we want to go down. Are you trying to figure out how changing the average of one measurement affects another? In other words, you're trying to make some relationship out of your data. So here, one measurement, I think a more accurate way to say is one variable. So let's say you have gene 1, that's one variable, another is another gene. If you want to study how the expression change in one gene affects the expression of another gene, then we go down. So otherwise we go left. So let's say that we go left first, okay? So you're not trying to figure out how changing the average of measurement affects another. So then here the question is, are you trying to predict measurement for individuals? So in other words, here, you're not trying to say something, so I think this question here is not very crisp clear as it is, but I want to add my own interpretation. So if we go left, we are going to say that basically we're going to say a general trend behind our data. So it's not about what we see, but we want to go deeper into what we see. So we're not talking about individual data points, but we're talking about the data set as a whole thing. So over here, we actually have two directions. One is called inferential, one is called predictive. Or maybe I can go back to clarify this a little bit more. So here, probably the easier way to say is that this is about causal relationship. This is about non-causal relationship. That's better or easier to understand. So this is association. This is causation, causality. So if we're only talking about the relationship between two variables without thinking what causes what, it's just an association, then we go left. Otherwise, we go down. So association is the primary target in statistical analysis. For most data sets, if we just have a snapshot of a state, like say single cell gene expression data, we just measure all the genes in all the cells together without having the previous state or the next state. So we essentially cannot say much about the causality. We can only say about association. So we are on the left side. And even for association, we can do statistical inference or prediction. So traditionally, people think statistical inference is the core of statistics, while prediction is the core of machine learning. That's the traditional view. So you can see the difference between the two. And why do we say they're different? The thing is that for inferential questions, you are more interested in the model parameters. In other words, for example, linear model. In the linear model, the parameter we are mostly interested in is the slope, right? The coefficient of the predictor. And that is a model parameter. We don't know its truth, and we can never observe the truth, but we want to infer the truth. So that's why we call it inferential. So inferential by nature is kind of a guess. You want to guess the model parameter given your data. And the underlying assumption is that you think your model is reasonable. Otherwise, the model parameter would not be meaningful at all. So inferential is concerning model, while in predictive questions, we care more about the thing we predict. For example, in the linear model, if you want to predict your y, y1 to yn, as we write as y1 hat to yn hat, then in that case, the prediction objective is to make your y1 hat to yn hat close to y1 to yn as much as possible. That's our goal. And the model parameter is less of a concern. So when people go for this route, they can usually accept complex or so-called black box models that are not really interpretable. For example, deep neural network. That's a very typical or extreme example. So in that case, people try to justify that model is reasonable by the prediction performance. See, the y1 hat to yn hat are really close to y1 to yn. That's the justification, but not the model parameters. On the other hand, I would say linear model is a typical simple model that usually do not predict very well, but it gives you very good inferential property. In other words, you can talk about the model parameters. You can say, okay, given my data, I estimate my parameters as those, and my estimates, how accurate are those estimates? I can quantify the accuracy of my estimates using, say, confidence intervals, or I can do statistical tests. So those all belong to the inferential part of the question. So usually we cannot satisfy both. In most cases, we have to either go for the inferential as our primary question or the prediction as our primary goal. So I would say in medical data analysis, the first question, if it is a regression type of question, the first question you want to think about is that, do I care more about the model parameters for interpretation, or do I care more about the prediction as my goal? So based on my experience, I think many questions are inferential in scientific research because we want to make some stories out of the data. And prediction is not so much of our concern in many cases because we have measurements for y1 to yn anyway. So if we want to predict a new data points yi, we usually trust our experiments, right? We want to measure that yi instead of predict it. But still, in some cases, if it's still not easy or too expensive to make new measurements, then predictive models may apply. For example, I know there are some work in using a machine learning model to predict open chromatin, or to predict transcription factor binding sites, which can be measured by ChIP-seq data, but it's quite expensive because you need to do ChIP-seq for every transcription factor, which is a different protein, and for every cell type. So if you cover all those experiments, that will be a lot of experiments to do, quite expensive. And for some proteins, you do not have a good antibody to do the ChIP-seq. So you can not easily have the data. So in that case, I know some people propose to use prediction to predict transcription factor binding from gene expression data. But biologically, it may not make that sense because we know, okay, gene expression is a downstream step of transcription factor binding. But still, there are computational work like that. So you can check them out. So that's kind of a computational problem in the bioinformatics field. And so if we go to the causality, this is not usually something statistics can handle, but still we have causal inference. That's kind of a subfield in statistics, and it has become more and more popular nowadays. But I have to say that causal inference is trying to infer causality from observational data without doing experiments. So you do not really observe the causal thing, but you try to infer causality by using your own assumption. So this is very common in social research because you have some common sense knowledge to build that causality graph. So that's what you assume. And given that causality graph, for example, smoking can lead to cancer, and then you want to infer the strength of the causal effect. So that's called causal inference. And finally, this one is something more fine. This is the finest level of modeling. It's called mechanistic modeling. And or it can be say deterministic modeling. So this is what we see in physics. If you think about Newton's laws, they all belong to those kind of models. So in other words, you know that if I change one thing by that amount, then the other thing will change by that amount for sure. So that's kind of a deterministic law. But in biological research, we usually do not have the luxury to go for a mechanistic model. I know in systems biology, people use mechanistic model like partial differential equations to study, for example, the relationship between transcription factor binding and gene expression. but that's usually at a very fine scale. So it's not really at the macro scale, but it's usually about one protein and one gene. So people can build models for that. But even in that case, they still think it's an approximation of biology. It's not a real biology. So one thing I want everybody to keep in mind is a very famous saying from a statistician George Box. The saying is that all models are wrong, but some are useful. So we should keep that in mind. And another thing I want to challenge, like a common wisdom, is that statistical methods or machine learning methods are unlike experimental techniques. So we can say, okay, probably like the next generation sequencing is generally better than microarray. It can give us more information about the data. But for statistical methods, it's not as clear as that. So for example, you cannot say deep neural network is better than linear model. They just have different strengths and they are suitable for different situations. So for example, deep learning requires you have a very, very large sample size. By sample size, I mean large number of data points to train the neural network because its model contains a lot of parameters. And you cannot have the parameter number bigger than your number of data points. So sometimes when your data points are too few, you cannot really afford such a complex model. And also, if your goal is to interpret model parameters, then the deep learning model doesn't fit your goal. So for those reasons, we cannot easily just say one model is better than that. But we can say, okay, maybe for my data, for my question, this model is more suitable than the other model. So that's one thing I want to make sure everyone understands. Because I was on some committees of bioinformatics students, and I saw that since the deep learning has become so popular, some students just want to try deep learning for their problem as the first attempt. But that's not usually well justified, right? You have to justify why you want to go to deep learning. Did you try simple model and they don't work well, right? I need to see those justifications for your modeling. Okay. So that's the first paper I want to talk about. So you can check the details later. And the second paper I want to talk about is this. This is something written by several famous statisticians for biologists. So I'm going to show this paper. It's called 10 Simple Rules for Effective Statistical Practice. And we will go through the rules quickly. So the first one is that statistical methods should enable data to answer scientific questions. Okay, this is aligned with that first science paper. We want to connect scientific questions to statistical method, and that's the goal of this class. We want to help you see the connection better. Okay, and the second rule is that signals always come with noise. I want to emphasize this point because this is the strength of statistics. So why do we want to do use statistics instead of just running some data summary? For example, everybody can calculate the mean, right? The average or the standard deviation or fit a curve from the data. Why isn't that enough? The reason is that those approaches are deterministic. They are treating data points as they are and try to make sense of the data points as they are. They do not think the data points carry noise with themselves. And what statistics does, and why statistics use probability theory as its backbone, is because statistics think data points are some random measurements. They come with noise. And the probability distributions like Gaussian, Poisson, binomial, they are used to model those noise. So if you think about that way, then you can see, okay, the probability distributions are not just used for some fancy thing, they are used for real purposes. We use probability distribution to describe what we believe that relationship should be like between data and the true signal. So for example, if you assume the data follows Gaussian distribution and the Gaussian describes the noise distribution, then the signal would be the Gaussian mean. And anything different from the mean would be what you believe as noise. So we want to carry this mindset in our data analysis. There's noise. And in my own research, I just want to make a side note. In my own research, if you're interested, so I had a paper from the 2015 in the science paper. So in that one, what we did is that we reanalyzed a 2011 Nature paper. And that Nature paper is about the relationship between 4 steps in the central dogma, including transcription, translation, mRNA degradation, protein degradation, and how much do those 4 steps contribute to the protein abundance levels we observe in cells. So in that original paper, the conclusion at that time that was in 2011 was quite shocking. They said that translation plays a more important role than transcription does. So that challenges a lot of the classical molecular biology results, because in the old times, we do not have very good quantification technologies for proteins. So a lot of the discoveries were made at the mRNA messenger RNA level. So if that statement holds, that is, translation is more dominant than transcription, then those results will not become so meaningful, right? Because you believe the mRNA levels do not really reflect protein levels that well. But by reanalyzing their data, we found that the original paper ignored the uncertainty or noise in their measurements, in both mRNA measurements and protein measurements. And by accounting for those measurement randomness, we found that indeed, at least based on their data, transcription is still more important than translation. So that case actually reflects how different statistical analysis may change our scientific conclusions. And rule 3 is plan ahead, really ahead. I think this is a rule for people who do experiments. Maybe some of us here will do experiments. So I think that the take-home message is that before you design your experiment, if you think you need to do statistical analysis after that, maybe you should consult a statistician to make sure your experimental design is reasonable. So that can save you a lot of time for your downstream analysis. For example, we should consider how to effectively reduce batch effects, how to reduce the correlation between experiments. So those key things will make the downstream statistical analysis more powerful. And rule 4 is about worry about data quality. So this is related to the rule 3. So if the data has been obtained already, you need to make sure the data has good quality, it's not biased, it do not contain too many artifacts. So for this one, I would say it relies on your expertise. So your domain knowledge is very important. You need to check your data very carefully to make sure, okay, the signal you have there is really signal, it's really biological signal, it's not some artifact. So for example, I think I heard a talk by a speaker before. So he said that he's a statistician, and when he collaborated with biologists, they found that one sample looks very different from another sample. They're doing like control experiment comparison. And they originally thought that's a signal, means that, okay, this gene is indeed differentially expressed from the control condition. But then they found that, okay, that signal was actually due to sample contamination. So you cannot know that without really working with biologists who know the data very well. So this is an issue with data quality. So I think probably this is not a strength of our bioinformatics people. To avoid that, we have to work with biologists who generate the data to make sure the data are good. And also you can use exploratory data analysis to check the data thoroughly to inspect any abnormal trends if you can see. And rule 5 is that statistical analysis is more than a set of computations. Okay, this is a very common misunderstanding that statistical analysis is just running R functions one by one as a pipeline, but it's more than that. So the most important thing is that you need to see rule 1, the one that the analysis really answers the scientific question. And also you need to make sure that you understand what each step is doing. So if you do not, probably it's a good idea to work with a statistician to make sure you fully understand it. Or if you are using a popular method, probably that's usually considered safer. But still, it's always better to know more about what the method is doing. And I think that's the reason why you're taking this class. So hopefully you can see, okay, why DSIG2, what DSIG2 is doing, right? That's one example. Okay, and also the rule 10, we'll see that later, is to make sure that you keep all your codes in a very readable and transparent way. Okay, rule 6, keep it simple. This is related to what I just said. And there's a more fancy name called Occam's razor. So it means that if we have several models that give us similar results, we always prefer the simplest model. So that's the keep it simple, what it means. So that's why I said when you try to use a predictive model, start with something simple until you see, okay, this model doesn't satisfy my goal. I want to move to something more complicated. And trying something very heavy and complex, I think that's just not so good training in the field. But hopefully I can educate you guys in this class to think about the beauty of simplicity. So try to think about if you can solve it using a simple analysis, that's usually considered more robust. So by being robust in statistical sense is that when you change your data a little bit, your results do not change much. And that's usually the case for simple models, but not so much for complex models. Okay, and also one more thing, just as a side note, why deep learning has been so popular in fields like artificial intelligence, computer vision, natural language processing. If you think about it, you can see the biggest difference between those fields and bioinformatics is that their data are much cleaner and their data have much larger sample size. So for example, in computer vision, I have a colleague who told me that they have images for every word in the dictionary. So there's no wonder, and those images were taken by high resolution cameras. So there's no doubt that, okay, we trust our data quality and we can evaluate the prediction results, right? We can see, okay, whether the classifier can classify this as dogs, this as cat. We can judge that by our human mind. But in biology, even researchers do not really know whether the data are so trustworthy or not. Otherwise, we will not call it scientific research. And a lot of times the data were generated for the first time. So in that case, we need to be more conservative in doing our analysis. Sometimes we need to step back a little bit, trying to say, okay, maybe we're not so sure about the quality. And I feel like statistics, unlike machine learning, is tending to give people more negative messages. Usually you use statistics to be less certain. You understand, okay, given that my parameter estimate has give me a very wide confidence interval for my parameter, then I will not trust my estimate that much. Okay, rule 7, provide assessment of variability. That's aligned with my statement about confidence intervals. So for every estimator you give, you want to also give a standard error for your estimate. And the standard error is the core part for construct the confidence interval. So that reflects how much you trust your estimate. And normally that standard error will decrease as your sample size increases. That means that with more data points, you are more certain about your estimate. But this is very important in statistical analysis. You need to provide that standard error, or you can provide a p-value. That's another thing we're going to talk soon, hopefully before the course, before this lecture ends. Yeah, so p-value is something that's so widely used, but so widely misused in bioinformatics. Okay, rule 8, check your assumptions. This is something we will provide you in this class. For the methods we give you, we will provide the assumptions. So then you will have this mindset when you read bioinformatics papers, that is, try to check the assumptions in the statistical method, check if the authors provide them. If they do, whether they make sense to you. And if a method paper didn't really lay out the assumptions, usually that's a strong warning sign. It means that probably the method section was not so rigorously written. Rule 9, when possible, replicate. This is about experimental design. So if your cost is affordable, try to replicate your experiment. That only having replicates can you do statistical analysis. If you have only one replicate, that's usually some challenging thing. But I will talk about this in a very soon short time. We'll talk about p-values. Okay, so rule 10, make your analysis reproducible. So this is about the habit of writing scientific papers. So in every scientific paper, we will have a method section. We will have a supplementary file. So those are the places for you to write out how you did your analysis in detail. Because your reader, if they want to replicate your analysis, they will check those parts to make sure they can really follow your code to reproduce your figures. So now we have those R Markdown or IPython notebooks for us to do the code, to do the reproducibility coding. Okay, so this is the 10 rules. And the next one I want to say probably is this one. So since we are running out of time, I'll try to be quick to give you the highlights of those papers, and then you can read yourself. So this one is actually a Nature comment. It's written by some Google scientists about 3 pitfalls to avoid in machine learning. Let's look at them one by one. The first one is splitting data inappropriately. So what does this mean? You know, in machine learning, a very common technique is called cross-validation, if many of you have heard of. So cross-validation means that you train your model on one part of your data, you evaluate the prediction performance of your model in another part of the data that is not used for training. So this can avoid the issue called overfitting, which means that your model just tend to fit your data very well, but it does not generalize to new data. So using cross-validation, you can avoid that. But if your data are split inappropriately, for example, if your test data do not come from the same distribution as your training data, then cross-validation will not work. That's the point. That's the point being said here. So the cross-validation idea only works when your test data are actually from your training data. So let me give you a counterexample. Let's say I train a predictive classifier on patients from one hospital in the United States, but I want to apply this classifier to another hospital in Africa. So if the two hospitals are not really comparable, their patient characteristics are not comparable, then for sure it will not work very well. So this is the thing we need to realize about the limitation of prediction trained based on machine learning algorithms. Another thing is called hidden variables. So in bioinformatics research, this is usually called as confounders. So hidden variables, let me give you one example, could be the batches. So if your data, say experimental data, come from 2 conditions, one is the treatment condition, one is the background or negative control condition. You compare the data by contrasting the 2 conditions. If your data were generated in 2 batches, the treatment in one batch, control in another batch, then the difference you see between the 2 data sets could be due to batch effects, not necessarily due to the condition change. So in this case, you cannot really entangle the 2 effects out because your experimental design does not allow you to do that. So how can you fix this problem? In experimental design, you may actually, if you have to generate 2 batches and each condition you need to replicate, for example, then you should have one batch include conditions, include both conditions. Say batch 1 includes replicate 1 in treatment and replicate 1 in control. Batch 2 includes replicate 2 in treatment, replicate 2 in control. So in that, you actually have the batch effect orthogonal to your biological effect. So only by designing experiment in that way, you can try to correct the batch effects. I'm just making up one example for that case. Okay, lastly, mistaking the objective. This is also aligned with the first issue we mentioned. Your analysis goal does not align with your scientific goal. Okay, so for this question, I want to refer you to read my own paper, which is also in that Dropbox folder. So I wrote a recent paper article about the difference between statistical hypothesis testing and classification, machine learning classification. So you can read that article to see the distinction between the 2 common analyses. So in fact, even though both of them can be used to address binary questions whose answers are yes or no, but they are talking about different objectives. So just in short, maybe one sentence summary for that paper is that hypothesis testing is trying to answer questions related to model parameters, which are not observable. In contrast, classification are trying to answer questions regarding individual data points. So you may observe the answers if you have new data with the labels. So that's the fundamental difference between the two things. And a related conclusion is that you use hypothesis testing to answer questions related to your features. And you use classification to answer questions related to your data points, or we say observations. So maybe we can defer the questions for you guys to ask me in the beginning of our next lecture after you read those papers in detail. Okay, so that's what I want to say about the 3 pitfalls. So it seems that I'm giving you a lot of negative messages in the first class, but I want to give you the warning because I feel like statistical analysis in many practices are just, people are too confident with their analysis, not too doubtful. So I think being doubtful is better than being confident, because at least you are trying to give your readers some room to doubt. Okay, so the scientific conclusion may not be 100% sure. So finally, I'll finish this lecture by talking about p-values. So p-values are just the tip of the iceberg. Maybe we can look at the data pipeline again. This is also written by Jeff and Roger. So basically, our analysis or pipeline has so many steps, right? You have experimental design, and you collect the data collection, and you obtain the raw data, and you clean the data, you tidy the data, right? So for example, in single cell gene expression data analysis, a necessary cleaning step is to remove the so-called cells that do not have many genes expressed. That's usually considered as the empty droplets. We remove those. And we also want to remove the genes that are not expressed at all because they're not informative for our analysis. So after the cleaning, we'll have fewer cells and fewer genes. And we will do exploratory data analysis. So this includes checking the distribution of the cells' total gene expression levels and to check individual gene expression levels. So this is usually to make sure the data has reasonable quality. So for example, biologists usually are interested in certain genes, right? Certain famous genes, and they want to check the expression pattern of those genes. If they're not reasonable, then you know, okay, the data are not well generated. And you may also want to do clustering of your cells to see if the cell clusters agree with your knowledge about cell types. So those are typical exploratory data analysis. And then you will formulate your scientific question. What is the question? So mostly the questions are related to, say, I want to identify differentially expressed genes. Then that's the problem DE62 tries to address, right? They want to propose a statistical model to address that question. And the model will actually output some summary statistic. So for example, in bioinformatics, a typical summary statistic would be the log-fold change, right? If you compare the gene expression level between 2 conditions, you can calculate the log-fold change. But having the log-fold change alone doesn't give you statistical insights, because you know the log-fold change is affected by measurement uncertainty, like the noise. Then how can you say whether the gene is differentially expressed or not? To do that, we need to do a hypothesis testing. And that hypothesis testing is what we call inference. So if you have taken 100B, you know the core components of hypothesis testing. That's also in my paper about the statistical hypothesis testing versus classification. So you know statistical hypothesis testing requires you to specify a null hypothesis, which is like the default or baseline, and alternative hypothesis, which is on the other side. If you reject the null hypothesis, you usually think, okay, I'm capturing the biological signal. So that's the whole philosophy. And to reject the null, you need to have a statistic, right? That's the summary statistic. And you need to have its distribution under a null hypothesis. So given the statistical value you observe, given the null distribution, you come up with a p-value. And p-value is something that's commonly mistaken. So probably we can leave this for you to think about. What's your understanding of p-value? And before you look it up, what's your understanding of p-value? And then you can check the Wikipedia to check the definition to see if it's aligned. But anyway, I want to say that p-value depends on the correct specification of the null distribution of the statistic. If the distribution is misspecified, then the p-value is misspecified. Then a lot of downstream analysis will be in trouble. So just be cautious that p-value is depending on your assumption. So for the same data, for the same scientific question, if you use a different statistic or if you assume a different data distribution, the p-value could be different. So it's possible to obtain multiple p-values from the same data set. And a bad practice is that people tend to pick the best p-value, which is the smallest p-value out of the multiple, and then report that in scientific paper. So that's the common misuse of p-value in scientific research. And that has led to what people call the reproducibility crisis. So we shouldn't do that after taking this class. And so we'll revisit this topic next time. So my take-home message for this time is that try to read the papers I send out. And you don't have to read everyone. You can just take your time, read the ones that interest you, and maybe come back with some questions to ask me in the beginning of our next class. So hopefully we'll begin from there. And then next class, we'll start with the introduction of gene expression analysis. That will be the technical part. Okay, so any questions before we finish? All right, so nice to see you today. So see you on Thursday.
Professor?
Yes.
I had a question. Is it imperative that we use R for the homework?
Sorry?
Do we have to use R? Can we use Python or something?
Yeah, you don't have to use R as long as you provide your code.
Okay. And when will you be giving the first homework to us?
So maybe I anticipate to assign the first homework maybe this Friday, and it will be due in 2 weeks.
Okay.
Yeah. Okay.
Bye.
Okay, bye.